Okay, PBio, what you are looking at is a simulation for an enzyme lab. Okay, just to kind of show you the bits and pieces of it, we have a key located down in the bottom right-hand corner. For the green ones are enzymes, the big green one. The substrates are blue, the products are red, the inhibitors are yellow. So if I was to, right now I have it set up with one enzyme, 10 substrates, 5 inhibitors, the temperature is 25 degrees, the container is 400, it doesn't really give a unit, so I want you to assume that it is milliliters, and the pH, okay? If I wanted to change the pH, or if I wanted to change the size of the container, I could. Okay, if you change the size of the container and you click and you push setup, then it changes the size of the container. Okay, and you will be you're not going to be changing the size of the container, but you will be changing some variables in this lab. There's actually going to be 3 different data collections, so you're going to have 3 different data tables, and you're going to have 3 different graphs. Now the graph paper, that is, if you don't have any graph paper, I have some available on the link that is below the button for this video. And you can download I'd like 3 pieces of graph paper. Um, the graph can be for each one of the experiments you're doing. The table can be one half, the graph can be the other half. Remember, these graphs are in pen and the tables are in pen. There's no pencil for this. All right? So let's say I was set up for 400, 25 degrees Celsius, pH of 7. My enzymes, substrates, and inhibitors are going. I push start, and they start moving around, or at least they should start moving around. There they go. So, as you can see, when the enzyme comes in contact with the substrate, it will turn it into a product. Okay. As the different experiments go along, you'll notice that the substrates will turn into products, unless, of course, you have an inhibitor there, and we will be looking at the inhibitors and the effect of inhibitor on the reaction rate. Okay. Now, on the AP Bio links page, there's a button that's going to say uh, simulation conditions or enzyme simulation conditions. Okay, and that will bring you to this page right here. Okay, so what this each table is doing, this is not a data table for you, it is just a table so you guys can see the different conditions for each lab. Okay, so there's 3 different experiments you're going to do. All right, so the enzyme substrate inhibitor temperature container, like I said, we're assuming it's milliliters, and pH is making reference to the enzyme substrate inhibitors temperature container and pH that is on this simulation. All right, so you can always stop it. Okay, in fact, for your rate, you will be stopping this every 30 seconds, and we'll get to that later. But you can change the conditions, and you can do a different setup um, as you go through the different labs. So, for example, the first experiment, you're going to determine the optimal temperature for the enzyme. Okay, it's the same enzyme for everybody and for all trials. So for each of the temperature settings below, because if you notice I have temperature settings of 10, 20, 30, 40, 50, and 60 degrees Celsius. Okay, I want you to have the simulation run for 1 minute. Okay, so you're going to want to have a stopwatch, or at least this clock with a second hand would be fine. Then you're going to determine the amount of product formed after that minute, and you're going to present that data in a graph. Okay, so you have your conditions set for experiment 1, so you can set it up and then run it. All right. Now, experiment 2. Experiment 2 is going to be very similar, but what you're going to do is use the determine the optimal pH for the enzyme. So for each of the pH settings below, which would be 1, 2, 3, 4, 5, 6, 7, 8, allow the simulation to run for 1 minute, and then determine the amount of product formed after that minute, and then present your data in a graph. So from both of the first 2 experiments, what you're really doing is finding out what the optimal temperature is. Or sorry, what the optimal temperature is and what the optimal pH is for this particular enzyme. Okay, now your third experiment. Okay, your third experiment, determining the effect of inhibitors have on enzymatic reaction rate. All right, so you're going to use the data from your previous 2 experiments to determine what the optimal temperature and what the optimal pH was, and you're going to enter those into the diagram, okay, or into the simulation. Okay, so once you have, based on your graph and based on your data, what the optimal temperature is, what the optimal pH is, then you're going to enter that in for experiment 3. Okay, the substrates, you're still sticking with 50. Okay, and then with the inhibitor, you're actually going to do 5 different trials, one at 0, one at 5, one at 10, one at 15, and one at 20. So you're going to have 5 different conditions for inhibitors, one with no inhibitors and one with the maximum amount of inhibitors we can get. And then your container itself does not change size. Now, your instructions, okay, use the data from your previous experiments to enter the appropriate value in temperature, in value for temperature and pH. Okay, you need a stopwatch for this because you're going to stop, okay, you're going to click the stop button on the simulation every 30 seconds for 240 seconds, which is 3 minutes. Okay, so if you have 5 different, this is roughly going to be a oops, sorry about that. It's 240 is going to be for 4 minutes, okay? And because every 30 seconds, you're going to actually have 8 pieces of data for every trial, okay? So each trial is going to have 8 pieces of data. You're going to have 5 different trials, one at each different amount of inhibitors, okay? So you'll conduct this for each of the each of the 5 amounts of inhibitors, and then present your data in a graph. So it is all 5, 0, 5, 10, 15, and 20. All right. Now, remember, it's every 30 seconds for 240 seconds for 4 minutes. So make sure you have a stopwatch so you can stop your clock. Okay. You're going to present all of the data from all 3 experiments in 3 different data tables: one for experiment 1, one for experiment 2, one for experiment 3, and then you're going to graph that data. And like I said, make sure that your graphs are in PIN. If you have any questions, make sure you ask in class, or you can shoot me an email.